with the CWD isolate, and there's no staining of the isolate in this, indicating that the Spiroplasmarum is immunologically distinct from the Spiroplasma TSE isolate. And in this culture here, we can see the organism by electron microscopy in the culture. This could be a very interesting cell model to look at, resistance, etc. That was mentioned. So here is the electron micrograph showing the organism in that infected cell culture. Now, spiroplasma also produce biofilm. And what we do, we have the organism growing in broth culture. We take they love nickel wire. So what you do is to take the wire and put it into the, sterilize it and put it into the cell culture and leave it for a week. And then pull the wire out, wash it off, and look at it by scanning electron microscopy, where you're looking at the surface of the material. And here's some normal wire. Here is the wire that's exposed to the spiroplasma broth culture. This is with the isolate. Works both with the isolate and also the laboratory strain. But you can see the collection of biofilm on the surface. And here's a higher magnification of that biofilm with here you can see portions of organism. But basically they cover themselves with a polysaccharide, and they're living there in a moist environment. They're perfectly happy, and they can survive in this fashion on plant surfaces, etc. Now, there was a similar experiment done with scrapie. And basically, what this group did was to expose wire, stainless steel wire, to a homogeneous of scrapie material. And here they have, I'm not sure why, but they decided to look at it by scanning electron microscopy. And the normal wire and the abnormal wire, there's some changes here, but here's a higher magnification of this. And you can see this sort of thing. Now, it looks identical to biofilm that I've been demonstrated with spiroplasma. And, they say, well, this is just prion. Well, no, I think it's biofilm. And if you take that wire and lay on an agar plate, the organisms from that biofilm will grow out into the agar and form, see all these colonies migrating out into the agar. So all those surface plaques on that wire are viable and able to form colonies on agar. Yes?
And a protein would not do that?
I wouldn't think so. By the way, this material is prion-free. We've tested it. We've done immunoblots on the material, and there's no prion associated with it. Now, what's absolutely fascinating is these organisms, like clay. Here, this is forming spiroplasma, forming biofilm on mica, which is a clay related. But look at this change here. Here's an organism, and a lot of they could be in coccoid forms as well. Obviously, from what I showed you, they tend to form these big hotel-like structures. But look at, here's an organism, here's another organism, and you have these long structures between the connections. And actually, it looks like something being transported from one organism to another. Now, this is very important, because if an organism, a bacteria, gets into soil, it has to communicate with other organisms. And you can imagine a soil particle in the center here, but these little communications, so to form a microcolony in the soil, you have to have that communication. So I think this could be the mechanism you would see it in the soil. So what are the goals of my project? One, we're eventually going to have to determine the relationship to the prion, and I got some ideas. We're finding a lot about this organism, especially we've shown this organism is resistant to autoclaving. We can boil the organism for an hour. We can leave it in 10% formalin overnight, still viable. So, you know, it has all the features of that original description in the 60s. And I believe that the prion is just a manifestation of this, an amyloid that's formed by interaction of the organism. And so we're in the process of studying that. But basically, we're working on genome studies, and we've got some phenomenal data that just got in the last few weeks that raises all very interesting possibilities, which I can't relate to you now. I'll be publishing that in the next 4 or 5 months. But surely we should be able to determine strain differences, and obviously with strain differences you should be able to trace infections. You know, whether human infection is related. Well, there is a possibility, especially since there's some young people in their 20s that have come down with CJD in the West. The pathology of chronic wasting disease shows what's called a floccular amyloid plaque, of prion amyloid plaque. And that's not the usual thing you see in CJD and scrapie, but it's seen in bovine spongiform encephalopathy. And the new variant disease in England had those type of plaques, indicating that it's a more virulent form of the organism. Manuelides at Yale has shown by transmission, you know, say, of Creutzfeldt-Jakob disease, serial passage of that will produce identical plaques, suggesting that we are dealing with a very virulent form of the organism in the CWD. Now, in relationship to humans, Creutzfeldt-Jakob disease is a disease between 40 and 80 years of age. And if you see it in the younger patient, you start to worry, and that's why the bovine spongiform encephalopathy was initially raised so much interest. Well, there are reports now of several cases in their 20s from in the West suggesting, wait, we may have something there, but that'll all be worked out, I think, down the road. Now, we have to look at strain differences between, say, scrapie, Creutzfeldt, and CWD. Toxins are a possibility, and we have to look at that. We should be able to determine that from the broth cultures. We need to do more testing of susceptibility. Perhaps we can, you know, test. You know, right now, there's no antibiotics will affect this, except tetracycline. Okay, tetracycline, And you can inhibit infection in experimental scrapie in mice with giving tetracycline. But it's, as I mentioned, a bacteriostatic drug. But right now, there's no other antibiotic that works. And we tested that. I tested that with the new method. In infection models, animal infection models, we need animal infectious models to test the validity of possible vaccines or diagnostic tests, and that's ongoing goal. Tissue culture models, it may have some benefit in susceptibility studies. With the genetic, you can alter the genetics of these tissue culture. Maybe that's applicable to what the other group are doing. I think it's possible. And you can challenge the infection with the actual bacteria. You can really, I right now have a live diagnostic test where we can actually take a sample from a deer and grow into broth culture and actually produce colonies on the agar plates. So we actually have a live test, but it's not. I don't think that's as practical as this organism. We have it in culture. We can get antibodies. We can develop a diagnostic for hunters, for example. There's an immunodiffusion test that you can have just a pocket test, and they can take a drop of blood from the animal, put on that. If it precipitate forms, you don't want to even dress it. But if it's negative, take it home and eat it. And the ELISA is possible since we produce antibodies, and it's going to be a little more difficult in the controlling things in the wild. Obviously, injectable vaccine would surely is doable with this, and would surely be able to protect your captive animals. This would be important for zoos as well, because they have captive animals, including the big cats that are probably susceptible. And what about the cattle industry? You could essentially vaccinate the cattle for this. And obviously we can look at grass samples and dirt samples with this live test. So I've gone over most of this, but studying the epidemiology, we talked to Scott the other night, and he's an epidemiologist, and he could use, I think I'm trying to get a lot of institutions involved in this. The University of Minnesota, it's an excellent school, and if we can get a lot of schools involved in this, looking at this in this direction, I think we can get some of this done a lot faster in my little lab. So the prion theory is flawed, and I think it's been the biggest problem in moving this whole field forward. The breakthrough, if I hadn't been able to grow it in cell-free culture, I would also have been dead in the water. I wouldn't have done it. The CWD epidemic has to be checked, no question. I mean, it's going to not only destroy your industry, but also, the hunting industry in general. We need, as I say, we need more laboratories involved. Now, Laura Manuelides, I just saw on the web, she made a comment to the press, And this is, I thought, quite interesting. She said that transmissible spongiform encephalopathies have a classic infectious disease pattern, but many prion proponents seem to have little knowledge of classic infectious disease. And I think the problem is, and I've always proposed that we should have the microbiology field looking at this disease rather than neurologists or even neuropathologists. They don't think in terms of fundamental established principles. They ignore facts that do not fit with their belief. Scientific truth is not determined by decree. And there was a recent decree by the Wildlife Agency that the prion is the cause of this disease. I think they should back up a little bit and look at the data. So I've been on this slow boat here, and I think it's getting a little clearer where we're going. That's it.
Thank you. Is there any questions anybody has? Lori?
Yeah, how are you coming with funding?
How are you coming with funding to help move it?
Oh, throughout my career, I'm a neuropathologist, so I've been able to keep, with the lack of funding, and there's been quite a big lack, I've been able to continue the research on the side. So I was paid for looking at brain biopsies and muscle biopsies, et cetera, but I could still keep the research going. Now, the funding, you know, others are talking about big grants. My grant size is about 50,000. And basically, we've been able to, I haven't worked for a salary since 2005, but that's not been my priority. But really, what we need is more labs, more research done along this line. The big problem is PhDs. A young PhD coming out of a university needs funds. If he can't get funds, he can't do research with any novel ideas. And the prion people sit on all the, at the NIH, sit on all the review boards. So there's just no way you're going to get funding from the NIH. Recently, I put in a grant application to work with scrapie in sheep, actually confirming the Koch's postulates, et cetera. And it was a $2 million grant. Large animal research is more expensive, obviously. But I talked to the manager at the NIH, and he said, well, I told him that the head of the Prion Center is on the committee. And he said, well, you have to have your peers involved. Okay, so it was turned down, and they gave a rating of, you know, quality of researcher, 1 to 10. I was an 8, but anyway, didn't bother me too much, except that I got an invitation to sit on a panel at the NIH to review his grant. His was a $10 million grant. And basically, what he was doing is, I think, questionable, but immaterial. I offered to sit on it, but within a week, I got a call from the NIH saying I was kicked off the committee because I was biased. Anyway, no, the standard government funding, which you really depend on, is just not there for this approach. Yes?
Where's the publication on the postulates you went through for the deer?
The-
Where's that published?
The publication of what?
You said you proved Koch's postulates for the CWD in deer.
Right. Well,
Where is that published?
I've got the last article, 2017. I show that that I discussed that Koch's postulates.
Okay, what journal is that?
Journal of Neuropathology, Experimental Neurology. It's the main journal for the American Association of Neuropathologists. I'll send you some publications, and also of the 207 article. Yes.
I just wanted to clarify. You said that the organism was able to withstand autoclave as well as
Yes.
high temperatures. And all
It's not published.
were you specifically saying spiroplasm?
The spiroplasma isolate from TSE. Yeah, this is what I've grown out of CWD. You cannot kill it with autoclaving, regular autoclaving.
That's not published yet?
Pardon?
That's not published yet, you said?
No. No, no, but it will be shortly.
One more sec. Also, in the sheep that you, or the deer that you inoculated with spiroplasma,
Right.
and recreated the lesions, did you test the neurotissue for the prion protein?
I sent, we sent some material to Dr. Hall at Iowa, and he came back equivocal. So we'll see. I really have to set up other models and and check that out. But I assume that we're going to see some evidence of prion formation with this isolate. Yes.
Can you talk a little bit more about what the vaccine would do in hopes of?
Well, injectable vaccine should work in protecting a captive herd. If you have a captive herd, whether it's in a zoo or the cattle industry or your business, it should protect it. But we have to validate all that. I mean, we have to prepare the vaccine, then we have to test it out in an animal model, preferably a ruminant model, and check it out. Now, diagnostic, we desperately need to do an ELISA, where you can check your whole herd in an afternoon. Now, if you see a positive, then you can go back to this live test that I have already and take that animal and get material and prove once and for all whether there's any organisms, that you can grow out of it. So, but right now, for human disease, well, Creutzfeldt-Jakob disease, as I mentioned, is a terrible disease. But right now, you get a death sentence. My point is that if you have a test, you diagnose it early, then treat it with antimicrobials. You got some new things like phage therapy. My gosh, I mean, you may be able to actually stop the process. I don't know. You have to do the experiments. It has to be done. I can't predict. It has to be. The experiments have to be done.
Is tetracycline a possible preventative?
No, problem with tetracycline, it won't kill anything, and it will stop, put the brakes on this infection. But the problem, the cattle industry has been giving tetracycline from birth to death, and they're stopping it soon, or have stopped it. And in that sense, we may get a spurt in TSE infection. I don't know. We'll see what happens. But tetracycline, the organisms will just, once in vitro, the organisms will just, I did a thymidine uptake study when I was at University of South Alabama, and it stymied the infection. It was, the curve was flat. but as soon as it wore off, went through a regular growth phase. So no, I don't think tetracycline will do it. Maybe some big company, once you go in the right direction, will come up with a bactericidal drug. That'll be great. But you just need to have, be going in the right direction. And that's, if I accomplish nothing more, I want to change the world to go that way rather than the way it's going.
I have one question.
Yeah.
How do you test for spiroplasma? How do you look for it?
Well, right now, it will grow in the broth culture, and then you can plate the organisms. You can see it actually with you saw a picture. You can see it by dark field microscopy. They'll trail this all the way across the field, but you can plate it on agar plates. And the classic test for microbiology are colonies on agar plates. I mean, that's the gold standard for microbiology. So they'll form those typical subsurface colonies, which you can enhance with oil red O staining.
So do you use blood samples, or what do you take?
Well, we've not tested blood for organisms, but I would think the big thing on blood samples would be spin them down, get the serum, and then test that with say a ELISA test. So that would be the way to go, I think.
We have a question.
I just wanted to say thank you for
Okay.
your passion and you looking into this deeper and just in doing it even despite having funding at times.
Yeah, I guess most of you have seen that video from Pennsylvania, and this guy, this Dr. John Evelyn, has really stirred the pot, and otherwise there wouldn't be this attention to it. But basically, his idea and mine as well is that what we want to do is to get other universities involved and going in this direction. And a lot can be accomplished by more investigators, especially the young PhDs. Yes?
You mentioned that you talked to somebody from the University of Minnesota. Did they seem like they were interested in
Dr. Wells, I talked to him last night, and he seemed amenable to it. In fact, he said to me, he said, well, this is pretty easy. And I said, yeah, it's not rocket science, it's there. And basically, I hope he does do that, because I was quite impressed with him.
Well, we thank you for coming, and he'll be here after. If anybody wants to corner him here with some questions, you're more than welcome. So,
Sure.
thank you for coming.
Thank you.